let's now talk about three kind of separate, unrelated concepts, but they come up a lot in AP biology. The first one is called dynamic equilibrium. And I have written here, reactants leads to products. So hopefully you remember that kind of thing where in chemistry class you started with some reactants, and then they reacted with each other to satisfy those valence electrons, right, to break and form chemical bonds, and we made some products. Well, I'm going to tell you, this arrow is sometimes deceiving because we pretty rarely go completely from reactants, they're all gone, to products. But okay. What oftentimes happens is these molecules, they are, let's say my hands are two different molecules, and I'll go like this. So you have one molecule, that's the reactant. Well, that thing, if they're in motion, maybe I give it some heat, I put it over a Bunsen burner and it breaks apart. Now I've got the products. So I got reactants and now I have products right here. Well, what happened? Every once in a while, couldn't they just slam back into each other and form the reactants again? And the answer is yes. Now, most of the times, one of these sides is more favorable. We're going to go more one way than the other. And so it's not this perfect, like you always start with reactants and then you look in your cup afterward and all you've got is products, right? They kind of go back and forth. They bounce back and forth because remember, these are just molecules. They're just bouncing around, slamming into each other, and they might form this again, and then every once in a while they get so much energy they shake apart again. That's how molecular reactions work, or chemical reactions work. So to kind of illustrate this point about what's going to happen here, and as we let this reaction progress, so like for example my hands reaction, we could start here and then we break apart and now we're like this, and then but every once in a while we're going to go back together, right? But let's imagine we have thousands of my hands doing this, and it's more common to go this way than that way at the beginning. How is it going to end up? Well, we got this thing called dynamic equilibrium, which means it never really ends. That's important. This doesn't just end. It's not just done. There's always going to be some molecules being created, some product being created, and there's always going to be some reactants being reformed, going back and forth. So I use this analogy of a mainland and an island. Let's say that the mainland has 1,000 people, and that's really overcrowded. All right, so it's an overcrowded mainland with 1,000 people. I know, okay, it's really small, whatever. Well, then there's this island over here that could hold up to, let's say, 100. Well, what's going to happen? Well, at first, there's going to be this giant mass exodus from the mainland as people are moving over to the island. But eventually, we're going to reach capacity here, right? And we're going to have 100 people over here, and that makes 900 over here. Well, that's all that this one can hold. Can't hold anymore, and now 900. Now we're feeling pretty good about our mainland. So it basically stops, but it's not actually. It stops in number, meaning we're not going to wind up gaining any more, losing any from the island and from the mainland. So if we look at if we take a snapshot about this, if we take a snapshot at any one period of time, there should be about 900 people here, 100 people there. But is it possible that every once in a while, sometimes a person's going to move back onto the mainland? Sure. Sure, that's possible. And then, of course, that opens up a spot, and so one person can bop over there. Again, back and forth. Now, it's probably not, the rate isn't as strong as it once was one way, right? And originally there was nobody going back this way because there was nobody on here to go back this way. But what's going to happen is we're going to come to a point where we have a certain amount of people here, a certain amount of people here, based on the area, how much each place can hold. And the rate of immigration and emigration, they're going to be about equal to each other, right? For every one person that bops back to the mainland, there's going to be one person that bops over to the island, right? So the same thing can be applied to reactants and products. They can go back and forth. At the end of the process, right, when we've come to a point, a certain point, where it doesn't look like anything else is taking place, it is not that the reaction has stopped. It's that for every time there's a product being formed, there's also a reactant being formed. They just go back and forth. It's not that they're equal. It's not that we have equal amounts of reactant products, just like we don't have equal number of people down here on the mainland and the island. It's that for every one that forms here, there's one that goes back. We get to a point where the reaction rates are equal. The reaction rates are equal. That's dynamic equilibrium. Now, let's move to something like this. Structure leads to function. In biology, now we can go on a big scale. We can say like your heart. It has a structure that pretty perfectly seems to match its function. It can fill up with blood, it can contract, it can do everything that it needs to do in order to keep you alive. Same story goes with the molecules in your body. Their structure that they have oftentimes leads directly to their function. So, for example, if I have this molecule right here, try to draw those edges kind of sharp, and this molecule right here, I'll just draw this. I'm hoping you can see that I drew this molecule that has a perfect shape to fit into this receptor on the surface of this cell, and this one has a perfect shape to fit into that receptor on that cell. So it literally is like a key in a lock, that this one fits this one and that one fits that one. This one would not fit into that one, and this one would not fit into that one. Now, so your body does this all the time. It makes certain hormones and chemicals. Let's, for example, go with dopamine. Dopamine, we like to say it's a neurotransmitter, but it's also this feel-good chemical. It makes you feel good. It takes away some natural pain, or it takes away some pain just naturally. Your body releases it when it wants to get rid of pain and wants to shut off some pain neurons. And let's go ahead and say that is dopamine. Right? It's going to perfectly fit in here. Well, of course, what we've done in the medical field is we've engineered, and of course in the illicit drug field too, this happens, but we've engineered molecules that have that shape, that fits perfectly in there. Now that molecule that we engineer doesn't have to have that same size or anything. It could be like this. No, I didn't mean to draw that part. I should make this part of it the same size, if I can. But what I'm saying is that it could have like this. It could just be huge and gigantic and can have this giant bulge off the top of it, right, wrapping around here, and I know it just looks like a scribble, but it has this part right here that would still perfectly fit in there. The other factor that does often become important is the charges on there. So let's say this one there's this like, this has like a slight positive charge right there. Well, then over here, I bet there's a negative charge. And this, our drug that we make, better also have a negative charge right there. So shape and charge become really important when we're talking about biological molecules interacting with each other. We have an entire unit dedicated to how molecules interact and how cells communicate with one another. So this molecule is going to should very nicely fit into that receptor on that cell. Now, if, let's say, something goes wrong and the receptor shape gets changed, right? Maybe it's a genetic defect. Maybe this person has a genetic defect. Well, then, they're not going to be able to fit that molecule. This molecule is never going to get in there. That cell is never going to be able to interact with that or the drug that maybe we make. and it can't do its function. Many diseases are caused by problems like this, the wrong receptor on the surface, or maybe your body can't make the right shape in the chemical communication hormone or whatever it is. So there's an example of how structure can lead to function in living things, especially in the chemistry of life. Finally, we have radioactive tracers. Pretty straightforward here, but people see the word radioactive and they think to themselves, oh my gosh, it's radiation. It's gonna kill something, is what oftentimes people say, or it's gonna lead to mutations and stuff. And that's actually true. Radiation can kill cells and can kill things, but not always. Not always. Radiation can just be simply like a molecule. Let's go with sulfur 34. Sulfur 34. We could draw that one out if we wanted to. Or carbon 14. Right? These are isotopes of the very common elements that are out there, and they will slowly break down and turn into their more stable counterparts, like in this case, carbon 12. So what we do sometimes is we take and let's go with this person right here. Let's say a doctor is worried about cancer in this person. They might actually attach one of these radioactive tracers to a molecule of glucose sugar, right? So let's go with, I'm just going to do this. I'm going to just draw this molecule. This is going to be sugar. And then what I'm going to do is I'm going to put a little glowing piece attached to it. I'm going to attach that little glowing piece to it. So what the doctor can do is the doctor can inject this person with this radioactive sugar. Now again, that radiation isn't going to kill this person. It's not meant to kill the cancer. It's just a little thing, and we can detect as that radiation is emitted from here, as that energy is released from that little piece, we can detect it. So if we inject this person with all these little molecules of radioactive glucose, then what we can do is we can put this person in a scanner, and we can see where are those molecules of glucose going. You know, the doctors will know, hey, this is where glucose should go in the body. It should go to your muscle cells and to your liver, to normally, right? It should go to these normal places. But what if we see extra radiation showing up in a lung, right? So there's light radiation pretty much everywhere, or just in the normal spots, but there's extra radiation in one lung and not in the other. Well, oftentimes in cancer, this is something that'll come up later, cancerous cells, tumor cells are really good at sucking extra energy. They're really good at pulling extra sugar towards them because they're energy consumers. They suck up extra energy. That's actually one of the major problems with cancer, is that it's just stealing every bit of energy. You eat, but it's all going to feeding the cancer cells, and then they get more and more invasive. So what the doctor can do is say, oh my gosh, look at all that radioactive glucose is being drawn to this left lung here. And that could mean that person has cancer in their left lung. Here's another example. If, let's say, we want to know how this virus affects living things like a mouse. What cells does it infect? Because viruses infect very specific cells of the body. What we can do is we can radioactively label the DNA inside that virus. So when that virus is allowed to infect this mouse over here, we can look and we can say which tissues are being most affected by that. And we just put that mouse, just like I said before, in that special machine, and we'll be able to see where is it glowing. Right? Is it affecting its bones? Is it affecting its skin? Is it affecting its throat cells? Where is it affecting that mouse? And we can do that because we've attached these radioactive tracers so that we can look and we can see how that energy is being emitted from those radioactive tracers. All right, and that is the last of the chemistry stuff that you need to know for biology.